immunoglobulins. So you've heard me use this term before. The thing that y'all are most familiar with is antibodies. And in a lot of textbooks, if you're reading about this, they'll call immunoglobulin the molecule that's associated with the membrane of the B cell, but the secreted version they call an antibody. So there you go. They're basically proteins that we find in plasma, serum, and other body secretions, especially breast milk shortly after giving birth. There's a lot of a certain isotype in there called secretory IgA that is critical, very important for an infant to coat their mucosal surfaces with, because it's important in taking care of those types of mucosal antigens and pathogens. You've heard the term antiserum. It's basically, if somebody's been immunized or been infected with something and they've made antibodies against it, then there should be higher concentrations of that particular antibody in the serum, so it's called antiserum. And in fact, it used to be, they used to give antisera from, if you had a snake bite, rattlesnake bite or something like that, they will have given the venom, and we used to do this in Okene. We catch rattlesnakes, you milk the venom, and then they would immunize horses with them and get them to develop antibodies against the antigen, and then they collect the serum, and that would serve as an antiserum. Not a good idea, I'm just telling you. That, as we go through this, you'll find out that when you introduce antibodies from a different species, you actually develop antibodies against the antibodies, and then you have these anaphylactic things, and people's arms fall off and stuff like that. It's just really gross. So you'll see that a little bit later.
Great.
So immunoglobulins are these things found in the gamma globulin fraction of the serum. That's where they got the name immunity plus globulin. They're pretty similar in structure, although we're going to find that certain types of these form 2 together or 5 together, and you get pentamers, and they do some different types of effective function. They have shared amino acid sequence with other immunoglobulin molecules. But as we mentioned, this top part up here that interacts with the antigen, that's the highly variable region. And it's variable because the amino acid sequences all right up here at the very top of this chain and that chain are going to be different from one antibody molecule to the next. And we'll see how that happens. Antigen gets its name from antibody generating. Antigen is going to be a substance that can bind specifically to an antibody. This is where it gets a little tricky. Some people are very picky about what's an antigen, what's an immunogen, and all these types of things, but these are kind of the definitions that I go by. An antigen is a substance that can bind specifically to an antibody or a T cell receptor will bind to it. It's an antigen. And an immunogen is going to be a substance that can induce an immune response. At some point, we'll talk a little bit about haptens. Haptens are things that are recognized by antibodies, but they're not big enough to induce an immune response. So you have to attach them to something bigger to immunize with. And at the end of this, you'll see I'll talk about things that play a role in immunogenicity of an antigen. Allergens are the ones that elicit, are the antigens that elicit allergic reactions. Tolerogen, it would be a name given to an antigen that induced tolerance instead of immunity. I used to do this all the time. We were studying these cells called T suppressor cells, cells that regulated immune responses, and now they're called T-reg cells or T-regulatory cells. But what we would do is we would give an antigen in a big slug intravenously. That's not a good way to vaccinate something. That's a good way to induce tolerance against something. Lots of antigen in the bloodstream will cause the immune system to stop responding to that. So that's tolerance. That would be a tolerogen. And we'll talk briefly about superantigens. And this is an example. Toxic shock syndrome toxin number 1 is a protein produced by Staph aureus that, in the case as a superantigen, it can stimulate multiple T cells, a big fraction of T cells that are not specific for it, but because of the T cell receptor type and the MHC that it's reacting to, it can stimulate a response even though it's not specific to the toxic shock toxin. All right, seen this before, but this is, again, the membrane-bound version of this Y-shaped molecule on the B cell. It's resting. It comes in contact with this antigen. It becomes activated, and it does a few things. It proliferates, so we have more of those B cells. Some of them differentiate into memory cells, so we have them hanging around and will respond very quickly. No lag phase next time when we see this antigen again. They just start producing, they start the double process, which is basically differentiate some of them into plasma cells, make these antibodies, and get them out to what they're supposed to do. Here's the structure. And we talked about this already. There's four chains here, 2 identical heavy chains, the green, 2 identical light chains in the yellow. There are disulfide bridges that hold these chains together. And we're going to look at different types of these things. They're called isotypes because they're a little bit different in the bottom part of the molecule. From this top part up here can't change much because it has to have a certain structure in order to recognize an antigen. But this down here can change because it has an effector function. And by altering what's going on down there, we get different effects. Some of them bind to Fc receptors, some of them bind to complement, some of them do different things. And in the case of IgE, which I've already told you about with mast cells, they go bind an Fc receptor on mast cells right away, which is unlike any other isotype. But we'll see those in a little bit. So covalent things, we have some glycosylation going on, but it's going to be different as well. The antigen binding site, as I said, is that N-terminal part. And so N-terminal of the light chain, here's the C-terminal, and that's N-terminal of the heavy, and the C-terminal of the heavy chain. And so the N-termini of both these heavy and light chain molecules form the antigen binding site, and that's the very highly variable part, especially the far up part. Not this whole domain in red, but the very high part up there. There's a lot of conserved amino acids in the lower part of the reds. It's the upper part is where you're going to see a lot of variability. And the C-terminal part, as I've already mentioned, is involved in the effector function of the immunoglobulin molecule. For the most part, that is not active until antigen is bound. As the antibody is floating around, it's not an active effector molecule. When it binds antigen, there's a conformational change in the molecule, and the FC portion becomes active. It can bind a complement, it can bind FC receptors, etc. Except, there's always an exception, IgE. IgE doesn't have to bind antigen first. It can go bind an FC receptor on a mast cell and do its thing. You have a hinge region here, and I'm going to show you more detail about that, but that adds a little flexibility so that the antibody molecule, if the antigens are this far apart on the pathogen, it can bind. If they're close together, it can bind. This, I only show you, this is pepsin and papain cleavage. This is the biochemical reactions that were done many years ago to understand the structure. And the reason why I show you this is because it helps you understand why they're named the way they are, and also in some therapies that you'll be hearing about in the future. All right, so in the top we have proteolytic cleavage by papain, and what ends up happening is you end up with 2 what are called FAB, fragment antigen binding. 2 arms, and they're pretty much identical. They're identical with papain's cleaved. And so they have the one, the left FAB still could bind the antigen, the right FAB can still bind the antigen, but only one. And then you have the FC portion. If you were to cleave with pepsin, you actually keep the 2 FAB arms intact, at least on some of the antibody molecules.